This next presentation comes to us all the way from Singapore. Dr. Prakash Kumar will walk us through his research on mangroves being conducted at the National University of Singapore.
Hi, good day to everyone. And let me begin by thanking Dr. Dharam Sharma and the organizers for inviting me to share our findings on the mangrove trees and what we do with the lessons we have learned from those. Right. So just to share with you, my major research interests include plant hormone signal transduction and control of plant development. And the mangrove studies include specifically the mechanism of salt tolerance in Avicennia officinalis. And the findings from these studies, we try to apply these for rice crop improvement, and especially the traits that we're looking for are salt tolerance and yield enhancement. Obviously, these are from the findings from our mangrove studies. And most of you are aware that abiotic stresses include high soil salinity, drought, flooding, etc. Right? These inhibit plant growth and yield, and it's estimated that salinity is a major problem, and about 2,000 hectares per day of irrigated land is affected by increased soil salinity. So this is like a huge area. And again, the lost crop value per year is estimated to be a staggering $27 billion, right? So the issue is that in irrigated land, how does the soil salinity increase? When you water it, doesn't the salt get washed away? So the issue is that continued irrigation brings in water with dissolved ions, and then water will evaporate off selectively, leaving behind the ions. So every season, when you keep on doing this, the salinity level increases. This is a really major problem, right? And so what we need to do is find ways to improve crop plants that are able to withstand this kind of stresses. So they are stress tolerant varieties, right? So it's important to do this. And what are some of the ways of doing this? Say, one way is the so-called pathway engineering strategy, right? So you find, say, the genetic pathway that helps the plants to withstand higher soil salinity, and then see if we can engineer it. So what are some of the possible pathways? Well, plant hormone signaling is an important pathway, and ion balance in the cells is affected by salinity and drought stress. So one of the things that we need to do is to use the so-called big data, right, and derive strategies for breeding crops with higher tolerance. So how exactly can we try and confer salinity tolerance to crop plants? Like I said, you have to use big data, which is the systems level or omics data, right? So you can get transcriptomics, genomics, phenomics, metabolomics, proteomics, all the omics you can think of, identify key genes responsible for salinity tolerance, and utilize this knowledge for improving salinity tolerance in the crop plants. So where exactly can we get such useful data from? All this omics data, where can we get it from? Well, the information from naturally salt-tolerant crop plants, or rather salt-tolerant plants, not necessarily crop plants, because we don't have many of these crop plants that tolerate so high salinity, right? So plants such as the mangroves can yield this kind of information if we do a systematic study. So how can we get that? Well, the vast majority of the plants are so-called glycophytes, or they're salt-sensitive types, such as rice and the laboratory plant Arabidopsis and all the brassica vegetables, etc. Right? So there are the salt tolerant varieties. These are examples for these are the mangrove trees, such as Avicennia officinalis, right? And they exhibit natural desalination process in them because they have these traits, such as efficient salt filtration at the roots and salt secreting glands on leaves. So just a bit of background. How exactly this is the plant propagation group, right? Propagators Society. So just to give you a little bit of the background, we don't do active propagation of mangroves by any artificial means. So it's all done by natural propagation for mangroves because even things such as tissue culture, mangroves are not amenable for it. So far, we don't have any protocols that are suitable for this. The tissues are laden with phenolics. The moment you put pieces of mangrove leaves or any plant material on tissue culture medium, it just turns brown. So the natural propagation occurs by means of fruits, or what we call propagules, right? Usually these are viviparous, right? That is, they germinate on the mother plant itself. As you can see, some of those that are hanging here, those are the propagules, right? So this is one particular species. This is the Bruguiera species. You can see the fruit. It has already germinated and a long hypocotyl-like structure is already there. So when it falls to the muddy ground, it can strike the tip buries itself in there and then it establishes itself as a seedling, right? There are other species of mangroves that have propagules that can float and then they're carried to long distances in sea currents. One of our earlier studies, published in Journal of Biogeography last year, is about the genetic structures across a biogeographical barrier reflect dispersal potential of 4 Southeast Asian mangrove plant species, and these are the species that we used: Avicennia, Sonoretia, Bruguiera, and Rhizophora, right? So this is a part of the study sites. This is where Singapore is, right at the tip of this peninsular Malaysia in Southeast Asia, right? And this is the Indonesian island Sumatra. That's off the coast of it is where the big tsunami struck a few years ago. So that's the location. Anyway, so what we have done is looked at the mangrove propagules that can float and then they're carried to long distances. And this is part of the Southeast Asian region, and that's Malaysia and that's Singapore again. This is the Indonesian island Sumatra. So these are examples of the different propagules. They're of different shapes and sizes. This is the Rhizophora, a long propagule. These are Bruguiera. This is Sonoretia and Avicennia, right? So these can float and establish themselves in long distances. Okay, so what is the desalination that occurs in mangroves? I mentioned they have natural desalination ability. What is it because? Or how did they acquire this? These are plants that literally have their life in a pickle situation. Get it? Life in a pickle, right? Plant tissue in salt water, right? So they inhabit their natural environment they inhabit is saline environment. They are constantly exposed to high seawater, right? High salinity in their environment, right? But they thrive in the marine and estuarine shorelines, right? Normal plants, you put salt water, seawater in them, within a few minutes they'll start wilting and they'll die. What are the special adaptations exhibited by mangroves that helps them to grow in such a hostile environment, high salinity? So There are two major adaptations I would like to highlight. First is the roots carry out ultrafiltration of salt, that is to prevent salt uptake. That's one strategy. But there's high amount of salt in there, and therefore some undesirable amounts of salt will get into the system, into the plant. So some mangroves that have this happening, which is despite their ultrafiltration, excess salt gets in. They have evolved a mechanism to secrete this at the salt glands on leaf surfaces. So these are the natural desalination processes at work. So these are like off the coast of our, just in our neighborhood within the camp, not within, just outside of the campus. Again, this is another view of that. So these are the salt crystals on leaf surface here on the bottom left, right? And the close-ups of the leaf sections in the middle here labeled salt glands, right? So there are two salt glands you can see, and the purplish stained section here, it's a close-up of the salt gland, right? So this is these are microscopic structures located on the leaf surface, and their roots are also able to carry out ultrafiltrations, right? So if we can understand how these two adaptations work, then the mechanism underlying this salt tolerance process, then that can yield the data we need to try and develop salt tolerant crops. So one of the ultrafiltration mechanisms is a physical mechanism, right? So as many of you are in the field know, there are two pathways of water uptake: the apoplastic route, which is through the porous cell wall, all the way to the endodermis in the root, and there it gets converted to the symplastic route because of the apoplastic barriers here, specifically lignin and suberin barriers on the endodermis cells, right, cell walls. So what is forced to go through the membrane, and that's what enforces the filtration effect. The second one is symplastic route, so it's through the cells, the interior of the cells, so it's with the filtration effect in place. Right, so one of the studies that we had conducted, we were able to demonstrate that there is this suberin deposition in the endodermis cells that helps to regulate ultrafiltration in the mangrove roots, right? So this is the control material. So this is the Bruguiera roots, and they have a uniformly suberized endodermis layer here right from the start, whether you give it salt or not. Right, so Avicennia seedlings that we have grown in the laboratory without exposure to salt develop very little of the suberized cells in the endodermis, whereas the moment you start giving it salt, this is salt treated on the right, this is control on the left. So the moment you start giving it the salt stress, so they start developing suberized layers, right? So we have published this in several publications. So what did we aim to do? So we aimed to understand the membrane proteins, how they function, identify them. What are these proteins that we're interested in? Mainly the transporters, the so-called aquaporins, or the water transporters, and ion channel proteins, right? So these are involved in salt uptake and secretion process, both at the roots and the salt glands at the leaf. And then we attempted to use biotechnological approaches for generating salt-tolerant plants, right? So first we did a study of differential gene expression. So we had the seedlings and treated with salt. So we had some that are treated, some that are controlled, extracted separately the RNA pool from these two batches of plant material. These are the Avicennia seedlings. And then we carried out the RNA-seq analysis, RNA sequencing, and then we looked for the differentially expressed genes. So the idea is which are the genes that are down-regulated and which are the genes that are up-regulated when we treat the seedlings with salt. And the idea is to see if we can identify these ion transporters that are involved in various essential tasks such as uptake of nutrients. Some of it is essential, potassium and nitrate, right? And if there's too much of sodium chloride, it will get in, right? So they're involved in controlling growth and then plant-water relations as well. So, just to summarize the findings from the RNA-seq, what we did is we did the computer analysis and compared it with the so-called KEGG pathway. This helps us to identify genes for several our desired biosynthetic pathways in the plants, right? So we looked for those that are involved in regulating suberin biosynthesis, and those that are differentially regulated were identified with the green and red boxes, right? So as you can see, there are several so-called CYP genes, CYP, that stands for cytochrome P450 group members, right? So 94A1, 86B1, they're all the different members within the cytochrome P450 grouping. And several of these are required for suberin biosynthesis, as mentioned on the left here. So these are some of the genes that we identified, and we started looking at their expression in the Avicennia mangrove and also the laboratory plant Arabidopsis, right? And this particular one I'm highlighting in this study is the cytochrome P450 member 94B1, right? This is upregulated by salt treatment. I'm not going to bother you with all the graph details, but those of you who are interested in it, it is down here. Now, this cytochrome P450 member 94B1, what does it do? The protein it encodes is an enzyme, right? So this helps to catalyze the synthesis of so-called omega hydroxy fatty acids, which are the suberin monomers, right? So this is the reason why the gene expression was pursued further. The study on the gene was pursued further by us. So the expression analysis of cytochrome P450s in Avicennia and Arabidopsis was examined. So these are Arabidopsis roots, and we have carried out expression analysis by fusing the GUS reporter gene and the GFP, green fluorescent protein reporter gene, under the promoter of the gene cytochrome P45094B1. So wherever you see the signal, GFP or blue for the GUS, wherever you see the signal, that gene is active, right? So this is expressed in roots and is induced by salt treatment, right? So in the control versus salt treated, right? So you see quite intense expression when treated with salt. And it's the right seedling, pardon me, it's the right part of the root, that namely the endodermis cells, right? So as you can see the close-up here. Now, this is preferentially expressed in the endodermis under salt treatment, as you see here. And like I mentioned already, it helps to catalyze the biosynthesis of a suberin monomer, right? So what does it mean for a plant? I mean, a gene expression is fine. So what we looked at was the seedlings and how the root length is affected by salinity, right? And in the adult plants of Arabidopsis, it's much easier to handle this Arabidopsis than the big giant mangrove trees, right? So we obtained the Arabidopsis mutant where this particular gene has been selectively knocked out. So this mutant, where the name is given in lowercase, right, ATCYP94B1, the mutant has this phenotype of the root length being inhibited when we treat it with salt, right, compared to the wild type, right? So compared to the wild type, the mutant's roots are inhibited. Now, when we overexpress this gene, in the mutant background itself, these seedlings are even more tolerant than the wild type. But without any treatment, there's no difference in phenotype, right? So this is untreated, fairly mature plants, right? So these are the salt-treated plants. You see the mutant is showing signs of serious stress, whereas the overexpression lines, where this particular gene is overexpressed, even in the mutant genetic background, right? So those plants do even better than the wild type. Okay, so they show better growth under sodium chloride treatment conditions. Now, we examined the suberin deposition in the roots of this mutant and the complementation line, right? So we were able to show quite clearly that the mutant line allows the apoplastic uptake of the dye, right? Whereas in the transgenic lines, where we have overexpressed the functional copy of this gene, the uptake is inhibited because they have significant expression of suberin in the endodermis layer, right? That prevents the apoplastic load. So to see if this can be translated into a crop plant, we generated transgenic rice plants expressing the Avicennia officinalis CYP94B1 gene, right? So these are normal wild type plant seedlings and the overexpression lines, right? So at 6 days after, 3 days after, and 6 days after, you can clearly see the height of the wild type seedlings is shorter. That's the normal growth, right? And these grow much faster in the presence of salt in the medium. Okay. Now, the other important thing is under the control conditions, there's no difference in their phenotype, the wild type and the overexpression lines, right? When we do the salt treatment, you begin to see the difference. It's quite apparent that the overexpressing lines, that is, the transgenic plants, rice plants, where we are expressing the Avicennia cytochrome P450 gene, the selected member, they grow much better. And even after the treatment, during the recovery growth, again, they maintain this growth much better. So you can see it in the graph down here. So the key thing was that to see if this mechanism is similar to what we studied in our Arabidopsis experimental plant, and indeed we found that the wild type, the suberin layer here versus the suberin layer in the transgenic plants overexpressing the Avicennia cytochrome P450 gene is significantly higher. Right? There's also an exodermis in rice roots, endodermis and exodermis. You can see the exodermis also having higher expression of this. So the higher salinity tolerance of these rice transgenic plants expressing the mangrove cytochrome P450 gene correlates with the increased suberin, right? Suberin deposition in their roots, especially in the endodermis and exodermis cells. So this is consistent with the findings that we just discussed with Arabidopsis. Therefore, salt tolerance trait can be conferred to rice by introducing the selected cytochrome P450 gene, right? So how are these cytochrome P450 genes regulated? We identified a particular transcription factor, namely one of the Werke transcription factors that are involved in the regulation. Without going through the details, I just want to highlight that what gave us the idea to look for this Werke transcription factor is the gene promoter has Werke binding domains, right? So there is the Werke binding, Werke 33. So the gene promoter has several of those Werke binding domains, right? Werke 9 here. So with that, we went back to our RNA-seq data. That's the big data, right? So we went back and looked for specifically what we're interested in, and we found that Werke 33 is expressed or induced in a similar manner to the CYP94. And therefore, we tested whether this actually shows the similar phenotypes. And then we looked at the ATCYP94B1 expression in the AT-Wiki 33 mutants, and we see quite clearly that in the mutant, the ATCYP94B1 expression is also significantly reduced. Right, so this is the Werke 33. This is the CYP. Okay, so the legend explains that. Okay, so uh, we have shown that salt induces at a WRKI33 gene expression, and in the mutant AT-WRKI3 loss of function, AT-CYP expression is down. That gave us a very good indication that AT-WRKI33 probably regulates the AT-CYP94B1 gene expression. And indeed, with several lines of data, 3 independent lines of data, such as the yeast studies and then the chromatin immunoprecipitation studies, we were able to show that AT-WRKI33 indeed does this function. Excellent. And therefore, what we did was we did the AT-CYP94B1 expression in the AT-WRKI33 background, mutant background, but this time under a different promoter, 35S promoter, that is not dependent on AT-WRKI33 transcription factor. And indeed, we're able to show the restoration of suberin layers, and the mutants now overcome that mutant phenotype, right? So they gain salt tolerance. So the model that we are expressing is this, we are showing is that the AT-CYP or rather CYP94B1 gene expression is controlled by WRKI33, which is under salt stress treatment, right? And this helps in establishing the required hydrophobic barrier formation in the right cells within the roots that helps to confer salt tolerance. Right, so this is the second last slide there. Just to summarize then, we have demonstrated that ultrafiltration in the mangrove Avicennia roots helps to exclude up to 95% of the salinity from or salt from the seawater. Right, the endodermis and exodermis layers show increased suberin in response to salt treatment, and several cytochrome P450 genes that help to synthesize the suberin are involved in this. They are differentially expressed, induced by salt, and we have identified a transcription factor. So what this shows is the molecular mechanism of this salinity tolerance. And using this, we have generated transgenic rice plants that express the Avicennia and also the Arabidopsis cytochrome P450 gene functional copy, and it helps to increase their salinity tolerance. The take-home messages, there are these 4 bullet points I want to leave you with. So the mangroves have special adaptations to survive in the saline environment, right? Namely ultrafiltration and salt secretion. And if we understand the physiological and molecular mechanisms of stress tolerance, we can then apply those findings to future crop plant varieties with higher abiotic stress tolerance, right? So we need to combine gene technologies with novel propagation and crop management techniques in order for us to be able to generate crop plants that can grow well with reduced inputs and that can tolerate increased abiotic stresses. With that, I acknowledge the funding from the Singapore National Research Foundation for our studies. And this is the happy group of researchers in the lab who have contributed to most of the findings that I shared with you today. And I thank you once again for all of you for attending the session and listening to my presentation despite the prevailing conditions globally. Thank you very much.
International sponsors like Harrell's and Ball. National sponsors, Nursery Supplies, Star Roses and Plants, Bailey Nurseries, Spring Meadow Nursery, ICL Specialty Fertilizer, and Bayer. And our most recent regional sponsor, I'd like to give a plug for the support of Van Bell Nursery.